to the microphone with questions or comments and input. And at that point as well, please introduce yourself with your name and your organization. Those who are attending virtually, there will be the Q&A for you to put questions in, and we have a person here on staff who will ask your question at the microphone. And so I think we're just going to give everyone about 2 more minutes, and then we'll get started for the day. So thank you. So first, I'd just like to say welcome, everyone, and thank you for joining us today. We are very happy and privileged to have this great turnout for today's meeting, those of you here in the room and those joining us virtually. As I mentioned a moment ago, my name is Nicholas King, and I am the Executive Director for the Predictive Safety Testing Consortium at the Critical Path Institute. As a brief introduction, I wanted to just kind of go through a little bit of what CPATH works on across all of our various projects and consortia, and then touch on this project objectives and then our objectives for today's meeting. Before I start out, though, I would like to give very nice acknowledgements to everyone who's helped in bringing this workshop to fruition today. First, I'd like to start off with the CPATH staff, who you can see listed here, as well as the FDA DARS staff, who have been our primary contacts over the past year as we've been planning this workshop. I'd also like to thank all of our presenters and panelists right now. And then as well, we formed work streams throughout this year, and everyone listed here, thank you very much for your contributions within those work streams to get us to today. And then I'd also like to just acknowledge the funding that the Critical Path Institute receives overall from the FDA, as well as from non-governmental sources. So a little bit about CPATH. CPATH was formed to address unmet medical needs, primarily in drug development, back in 2004. And today we do that through working in a primarily consortium model where we focus on some of our core competencies around biomarkers, clinical outcome assessments, data management and standards, modeling analytics, as well as regulatory and development science. One of our founding components of CPATH is that we've always maintained ourselves as a neutral third-party convener of multiple stakeholders. And through working collaboratively across within a consortium, we look for developing drug development tools as well as other solutions such as in silico models and databases of various disease areas to advance those models frequently. Currently, CPath is comprised of over 16 consortia as well as other projects, and we have focus in a variety of disease-specific areas, and then we have consortia that are disease agnostic, such as our clinical outcomes assessment program, as well as the predictive safety testing consortium. And so we, each consortium is comprised of frequently pharmaceutical companies, academic researchers, additional subject media experts within the respective field, working together towards new drug development tools or other appropriate solutions. And over the past, since 2004, in terms of regulatory successes, I'm not going to go through all of these, but you get a feel for the different areas where we've had successes. And within that, we've had, at the FDA, 8 qualification decisions, 7 letters of support, as well as 1 fit-for-purpose model endorsement. And then at the EMA, we've also had 8 qualification opinions and 10 letters of support. And as well, in Japan, we've had 1 qualification of the kidney safety injury biomarkers back in 2010. So that's just a little bit on CPath. And so, why are we here today? I think everyone hopefully knows that. But overall, this project, we have 3 primary objectives. And the first is to develop an evidentiary considerations framework for the qualification of complex in vitro models. I'm just going to call that the qualification framework in short, moving forward. Then agree on a liver complex in vitro model context of use to pursue for qualification and then initiate that qualification process. And third is to conduct studies to assess the performance of a model or models that will provide answers to the chosen context of use, and then assess that model based on the recommendations within the qualification framework. And so today we're here, we bring everyone, all of these stakeholders together, first to look at ways that we can refine the components of this standardized framework we've been working on to support the qualification of complex in vitro models for use in regulatory submissions. And second is to look at refining what we see as realistic and pragmatic DILI contexts of use to pursue for qualification. So really, everyone, I keep that in mind throughout the next 2 days. And so continuing on that theme, this is my request of everyone throughout the next 2 days. We're really looking for engaged discussion during our panel sessions. For the live audience, there's the microphones here. Again, please do introduce your name and your organization. And then the virtual audience, you can pose questions via the Q&A feature. Additionally, please do keep in mind the scope of this project's COUs in terms of application of complex in vitro models. And so on the right here, I have an image from a recent publication that's looking at the drug development life cycle. And we are focused on what's here circled in red, complex in vitro models that would be utilized to support an IND or NDA. And so when we're thinking about the context of use, please do keep that in mind, that's looking at, from the discussions we've had to this point, we've narrowed it down to not necessarily look at efficacy, but to focus in on toxicology and drug-induced liver injury. And so Through our agenda, everyone's seen this, but just to give a brief overview, we're going to start off the day with a few keynote addresses. The first is going to be by David Strauss, and from the Division of Applied Regulatory Science, and he's going to give a perspective on translating new drug development tools into regulatory use. Dr. Jeffrey Siegel is going to give an overview of qualification from his perspective as the office director for the office of from Otis. And then we're going to have two presentations from some excellent hepatologists, Dr. Mark Avigan and Dr. Eric Cohen, providing their perspective on DILI and opportunities for complex in vitro models in drug development. Following the break, we're going to move into our first session looking at developing contexts of use, and we'll have two shorter presentations and then a longer panel session and discussion. Following that, we'll have lunch and move into session 2. And in session 2, we're looking at analytical considerations for these models with two short presentations, followed again by a panel session. And then moving into session 3, the final one for today, will be looking at the biological performance considerations. And again, two short presentations followed by hopefully an engaged and engaged discussion. On the second day, we will have, we'll lead off the day with two more keynote addresses. First, Dr. Tina Morrison from the FDA will talk about some of the lessons learned in credibility assessment framework development and then context of use for computational models and where she sees applications from or learnings from that are applicable here with complex in vitro models. Second, Dr. Paul Skip Hayashi is going to provide his perspective as a clinician on the application of complex in vitro models in drug development and review. And then we're going to have a breakout session, and there our goal is to look at the three different outputs from the first three sessions on the first day and walk through a context of use that will refine and apply what we've assembled to date to consider what would be an appropriate qualification project outline for 4 different contexts of use. And so we'll split up into 4 different groups. Each group will have a COU and a moderator to facilitate walking through the steps. And then afterwards, we'll come back and report out on what that looks like. At lunch today, we'll post a form where you can sign up for the specific context of use that you would like to participate in tomorrow. We'll come back, summarize what the work was done during the breakout sessions, and then move into a final reflection on some refined context of use and close out with the next steps. So thank you, everyone. Next, I'd like to introduce our first speaker of the day, Dr. David Strauss. And David is the director of the Division of Applied Regulatory Science within CEDAR and has been in that office for several years. He has over 20 years of research experience and is going to speak to us today around his perspective on translating new drug development tools into regulatory use. David. The green.
Okay. All right, well, thank you, Nick, and thank you to the Critical Path Institute for helping us pull all of you together here, and really looking forward to having a robust set of talks and panel discussions and then the breakout sessions tomorrow. So, starting with why. Why do we need to move new science into the drug development process? This is a question that all of you should ask yourself, I think, and may have different answers to that question. I have some thoughts about this, and I'll get to that a little bit more as my talk goes forward. The next part that I'm really going to focus on most in this talk is the how and my perspective on how do we translate new drug development tools into regulatory use. And I'm going to draw on some prior experiences that are not in the drug-induced liver injury space, but in other areas. And then finally getting to what specific regulatory questions can new drug development tools help address today? And Nick put up a slide with phases of drug development. What we're interested in here and from the FDA's perspective is how we can focus on questions from the sort of regulated part of drug development. And I think there are some specific questions in context of use that are addressable today. So one of my, I'm going to draw on sort of two broader experiences. And the first, and I'm going to pull some information from a talk that we gave to the FDA Science Board a little over a year ago in June 2022, that was on the topic of advancing alternative methods for regulatory use. And we have a cross-agency group that is working on this topic and thinking about, not just in the drug development space, but across FDA, how we can advance alternative methods. And if we focus on the why here, there's a lot of promising new technologies. There's been advances in systems biology, stem cells, engineered tissues, mathematical modeling, and these present new opportunities to improve our ability to predict risk and efficacy. This includes microphysiological systems, combined in vitro and in silico models, and that is an approach we have taken in the cardiac safety space. Also, cellular models that can be genetically engineered cellular models. We've actually had applications in drug development where we've used cellular models to assess efficacy or effectiveness in rare diseases where we couldn't enroll, or the sponsor couldn't enroll, and we couldn't assess the effectiveness in certain variants, and we extended the indications of certain drugs based on cellular data. So these advances may bring products to market faster with improved efficacy or prevent products with increased toxicological risk from reaching the market. But how do we do that? And it's actually quite difficult, and multiple steps are required to translate these new technologies into regulatory use and maintain the same standards of safety, efficacy, and quality of FDA-regulated products. And I think the most important concept that I'm hoping more people think about at this workshop is the context of use. And it's sort of very challenging. It's still challenging for me, and I think you're going to hear more about that. You'll hear about it from Jeff Siegel in the next talk, I believe. And we need to have predictable models, reproducible models, quality control, and they need to be for contexts of use that are going to be useful in drug development and evaluation. And we're nowhere near being able to replace all animal testing, but there are opportunities for alternative methods to make additional inroads in addressing the three R's here, is referring to the replacing, reducing, and refining animal testing for specific contexts of use. And this was a slide that had been in that FDA Science Board presentation. So then coming back to the what, and so this image, there was a former member of our division that I had asked once, could he show an image of drug development, and he was a really good artist, and this is what he came up with. And so, the drug discovery and early development was sort of the top part of here. And I think in leading up to this workshop, there's various, from the model developer side or from the pharmaceutical industry side. I mean, this is a very important context of use, but it's not the context of use that we are most sort of in our wheelhouse at FDA. And we're really interested in the part after, or at the time of an IND, an investigational new drug application, but then phase 1 and 2 clinical development, phase 3 clinical development, the time of a marketing application, and you finally get a approved drug. And so when we say translating new drugs development tools into regulatory use, we're thinking about what are the questions we can help answer or address in those phases? So I was in my sort of scientific expertise and training for many years was in the cardiac space, cardiac safety, and I'm going to talk a little bit more about this cardiac safety initiative we had that really started 10 years ago. And then about 5 years ago, though, we opened the ICH guidelines, ICH E14 and S7B, which are the E14 is the clinical regulatory guideline for assessing pro-arrhythmic risk, and S7B is the non-clinical guideline. And they really were sort of completely separate documents. And you did the S7B studies, submitted the IND. If you're a drug company, FDA looks at it, and then we just like completely forget it. And clinical development goes on, and you follow E14. And there was like some suggestion in the guidelines, oh, they should like talk to each other, but it didn't happen. And so when we open, in the ICH group, which if you're not familiar is all the major global drug regulators around the world, and then representatives from industry, pharma in the US, the equivalent in Europe and Japan, and get together and see if you can harmonize regulatory guidelines. And in this case, we developed a combined set of questions and answers, which is then a new regulatory guideline in itself, of how to really integrate them together. And in this case, these are some slides that had been part of a webinar we did when the draft set of questions and answers had come out, and they were later finalized a year and a half ago. But the value proposition was they're directed at scenarios where non-clinical data can reduce the number of clinical studies. And so if you're familiar with clinical drug development, there's this so-called thorough QT study, TQT, and it's not very efficient and it's not that specific. And so can we reduce them? Streamline drug development, inform clinical decision-making at different stages during clinical development and also at the time of a marketing application. And how can you leverage the non-clinical data and the clinical data, for example, to better inform labeling, to better communicate risk. I mean, we have this like crazy language that gets into drug labels, and it's like no large QT effects, and it's like, what the heck does that mean? And I don't know what that means. But like, can we do something better and like communicate, actually this drug has low risk, this one, or we don't need to say anything. We don't, and so how can we have this better integrated risk assessment? And so how did we get there in that cardiac safety initiative? I mean, it was a large effort with many people at FDA, in the pharmaceutical industry, in the sort of CRO, contract research organization space, tool developers. There was applied research and collaboration, workshops, kind of like this one, developing white papers, and ultimately developing guidance, policy, training, and that's continuing. We actually still have a ICH E14 S7B discussion group. We had one of our calls earlier this week. I think it was Tuesday morning, and we're still figuring out how to implement and what the next steps are. But you may think, oh, great, we have a workshop. We'll just figure it out today and everything will go forward. But there were actually a lot of workshops. I mean, there were 5 workshops in this initiative, and there was studies going on in parallel. And then in 2018, that's when we opened the ICH guidelines to make the revisions that we did. But I think we can learn from this experience. And I'm not saying that this drug-induced, if we do something here in the drug-induced liver injury space, it's going to follow this model exactly. But I think there's some lessons that can be learned. And I think one of the lessons is like bringing all the stakeholders together. And because it's not like, oh, it's what somebody at FDA wants to do. It's everything's got to align to actually move some new process forward, in my opinion. And what were some of, there was a lot of applied research that went on in that initiative. And as this was not by any means the only study, but this related to an international, quote, multi-site study of human-induced pluripotent stem cell-derived cardiomyocytes for drug proarrhythmic potential assessment. And I mean, this was a study that FDA provided some funding to a consortia, but it really was a small amount of funding, and a large group of people from multiple continents and 10 laboratories were mainly volunteering their time to participate in this large consensus protocol driven study with different cell lines, different devices. And so it's something that happened there. Out of one of the final workshops, there were breakout sessions, and there were white papers that then were written over the following year on the left, white paper on human stem cell derived cardiomyocyte assays. On the right, on proarrhythmia model validation that was actually initially focused on the in silico models, computational models, but the principles were broad enough that they can apply to in vitro models as well. And I mean, you probably get the picture from the little infographics. We had a lot of people contributing, and these went on to then form the basis of some of what ended up in the ICH E14 S7B questions and answers. There's parts that are specifically to the clinical guideline, the E14, and then to the non-clinical guideline, S7B. And it starts out like the questions and answers to the non-clinical guideline is the first question and answer is all about the integrated risk assessment. But then other parts of the questions and answers, there's best practice recommendations for in vitro ion channel and human-induced pluripotent stem cell assays. And the last set of questions and answers to the non-clinical guideline is about principles for validating in vitro and in silico proarrhythmia models and qualifying them for regulatory use. And I think there's a lot of parallels that could expand, for example, to the liver space. So I just point these out if people are interested in the future. And we talked about that some in, there was an article that the FDA colleagues that were part of our ICH, group that representing the FDA for the E14 S7B working group. And there was a additional article that appeared in the same issue in Clinical Pharmacology and Therapeutics. It was highlighted on the cover. That was an industry perspective. And I mean, it mainly focused on the cardiac safety space, but talked about how this integrated non-clinical clinical assessment could serve as a model for other safety areas in drug development and regulatory evaluation. And so to wrap this up, why have this workshop? Well, as has been alluded to in the two talks already today, including mine, I think it's important to bring all the stakeholders together so the group can figure out the why, how, and what. So thanks for joining.